you've heard about repurposing sort of on and off throughout the morning. And you also heard that 40% of the ADDF pipeline is repurposed agents. So thinking about repurposing as a strategy for drug development is really important. And I want to give you some perspectives that I've developed relatively recently on this. So I'll start just by saying something about the Goods Prize and, of course, Nancy's great role in this. I'll define repurposing for you so that you know exactly what I'm talking about. We're in the city of the Nobel Prize, so I want to give you one example of repurposing as it relates to the Nobel Prize. Then I'll talk about a little bit about the pipeline strategies for repurposing in drug development, and I'll finish by talking about the ecosystem, which I think is an extremely important perspective about repurposing drug development. So start with the Goods Prize. Here's Melvin Goods. He was the CEO of Warner Lambert when Cognex was approved. So this was the first drug for Alzheimer's disease, Cognex. Rhoda was talking not long ago about, when we were up on stage here, about craziness versus innovation, and that margin can be blurred. And I am sure that when Melvin Goods proposed to the company that they should develop a drug for an untreatable disease, I'm sure that there were some people who lacked enthusiasm for that idea. He prevailed. And so the first drug for Alzheimer's disease was developed, Tacrin. It started the remarkable story. And for me, all of the drugs that have been approved since are part of that legacy, because the critical shift in opinion was Alzheimer's is treatable. Alzheimer's, until that time, had been regarded as untreatable. So now it became treatable. And then from there, of course, many more drugs evolved, including, drugs that had better side effect profiles or maybe better efficacy, the field matured from there. But this was such a critical first step because Alzheimer's disease became a treatable disorder. Now, repurposing then, just so you know exactly how I'm defining it when I use this, is the use of a drug that's approved for one indication for treatment of another indication. So I mentioned on the panel my own work with rasagiline. That is a drug that is approved for Parkinson's disease. And there were many reasons to believe that it could be efficacious in Alzheimer's disease. And that was the foundation for the trial that we did. Now, repurposed drugs have many advantages, and Howard talked a little bit about this. Something about their safety. And this is still the number one reason that drugs are stopped, is because a side effect emerges. So you have already handled that problem in a repurposed agent. The pharmacokinetics, that is, how fast does it get into the system? How high does it get into the blood, in the blood level? How long does it take to get to that high blood level? All of that is all solved by the repurposing, because you've already been through those early stages of the drug development. You know about dosing and manufacturing, and although we tend not to think too much about that in terms of drug development, it's really important, right? How you can get the drug to people if it were to be approved. So that's what repurposing is, and the rest of the talk will be devoted to that. Now, I mentioned that there's a Nobel legacy for repurposing. So here it is. So here it is. This is Sir James White Black. So a few things are black and white, right? But in this case, it's true. This is Sir James White Black. James Black won the Nobel Prize in 1998. for work that he had done with cimetidine, which is a very commonly used drug for gastroesophageal reflux. Very widely used. Some of you are probably on that drug or some drug like that. And it was based on his work with antihistamines, approved antihistamines. And he says during his interviews for the Nobel Prize, he says, the most fruitful basis for discovery of a new drug is to start with an old drug. And that's a pretty good definition of repurposing. To discover a new drug, start with an old drug. So that is the philosophy that led to the Nobel Prize of 1998. Now, I want to show you a little bit different take on the ADDF pipeline. And you heard that 40% of the ADDF pipeline is devoted specifically to repurposing. So here's the repertoire of targets that are represented in the ADDF pipeline. Synaptic plasticity, inflammation, the vascular components of Alzheimer's disease, bioenergetics, that is, can the cell use sugar and metabolize normally? Epigenetics, changes in some of the expression of genes in the cell. Neurogenesis, we've heard about Robbie's work with neurogenesis. Tau, proteostasis, neurodegeneration, and oxidation. And notably not on this list, for example, is amyloid, because I think Howard correctly perceived that there was a tremendous investment in amyloid through the pharmaceutical industry. And so the opportunity was to develop non-amyloid drugs. If we look then at this pipeline for the representation across the categories, you see that ADDF has been most invested in synaptic plasticity. Then with a very good investment in inflammation and in vascular disorders. So inflammation is such an important part of the pipeline in general and of the ADDF pipeline in particular. Then not long below that are bioenergetics, epigenetics, oxidation, and others. So you see that this is a very diversified pipeline that addresses many biological targets within the complications of Alzheimer's disease. Now let's look at ADDF's pipeline across the stages or phases of Alzheimer's disease drug development. So, of course, there's non-clinical development in animals in the laboratory. There's often some epidemiologic studies. Is the disease more represented in one group than another, or in one group of people who are taking one drug than another? This is a great segue to repurposing. For example, sildenafil or Viagra has been shown to be associated with a lower incidence of Alzheimer's disease in recent epidemiologic studies. So it's a good way to begin characterizing candidate agents. Relatively few agents in phase 1 or phase 3, and a big investment in phase 2. This is really important. These are proof of concept studies. They show whether the drug should be advanced into more late stage studies or not. This is an experimental medicine approach. This is something that Howard has emphasized many times. It depends on biomarker outcomes. They're smaller trials, so they tend not to have strong clinical outcomes, but they can have strong biomarker outcomes. And the analysis is more based on statistical structures such as effect sizes, directionality, and consistency rather than p-values, because you're looking for proof of concept. Is there evidence that this agent is working? And I'll just draw attention to a publication that was led by the ADDF, this 2021 publication in Neurology, about how to do phase 2 trials. And that's very important. You do phase 2 trials and you teach phase 2 trials so that people know how to do their trials well. And that publication is very important for that point of view. Now, new tools are available for repurposing, and I'm going to just show you a couple of things about this because I'm so excited about it. So in the upper left-hand corner, you see, for example, work on an example of network proximity. So a disease has a network. Let's say my right hand is the disease network with many connected nodes. Drugs have a network. So they affect more than one target in nearly all cases. So the question is, how close are these two networks, with the assumption being that the drug cannot work unless it has multiple nodes of interaction with the disease state. And this is now a numerically solvable problem. So network proximity, I think, is very exciting. Can do all sorts of animal validation and validation in human-derived stem cells now to show whether or not this drug that looks like it's good because maybe an epidemiologic study suggests it is, can you test that to see whether it is good in cell or animal models? And now we can use the electronic medical record. And that's, for example, how the work on Viagra was done. And that will tell us whether exposure to one agent is associated with either an increase or a decrease of Alzheimer's disease. So there are many new tools coming in to the scientific technology universe that can help us define repurposing drug candidates and be able to be more successful in terms of the eventual trial that is executed. Now, just show you one example here. So in blue is the network of Alzheimer's disease. And you see that there's many nodal nodes, and this would be nodes of amyloid, nodes of tau, nodes of inflammation, and so on, all the things we've been talking about today. Now, here are two repurposed drugs. Here's riluzole, and you see that it has an effect on some, but not all, of the nodes. And then here is carvedilol right here, and you can see that it has an effect on alternate nodes. So this suggests that the combination of these two agents would have the maximal effect on the nodes of the disease. So this is an example of proximity matching of two diseases, two drugs onto the disease process to discover candidate therapies and candidate combination therapies for Alzheimer's disease. Jacques asked this morning, how are we going to decide what drugs to test? Well, this is one way, Jacques, to decide what drugs to test. So there are some disadvantages to repurposing, and if we're really going to use this as a strategy, we have to overcome and we have to change the disadvantages. So it is uncommon for a generic agent, and most of these drugs are generic, to be advanced to phase 3 where they could become approved and therefore widely used because people would know about it and it would be manufactured on scale and make it available to people. That hardly ever happens because there is limited intellectual property on many of these generic agents. So if there's limited intellectual property, any generic company can make it, and the company that is advancing the compound cannot get a return on investment. And if you cannot get a return on your investment, you cannot advance that compound. So that's the great problem that is faced by repurposed agents. So the best intellectual property is called composition of matter, and this is already expired in generic agents. So what else can you do? And ADDF has been a pioneer in what else can you do. So you can do novel formulations, you can do novel dosing, you can do novel combinations, you can do use patents, such as this is for Alzheimer's disease as opposed to for Parkinson's disease, and you can do chemical modifications such as deuterization. So alternatives exist. They are not as strong as we would like them to be. And we have another problem. This is, I know this problem in the US, and I expect it is true in other countries as well, which is that Congress wants to move drugs from the branded state to the generic state so that people can get those drugs at the lowest possible cost. That's a good idea. But in so doing, the way they are changing the laws, it makes it more and more difficult to have any intellectual property associated with a generic agent. And therefore, it's more and more difficult over time to advance a generic agent into advanced stage testing. So one long-term strategy has to be to acquaint Congress with the downstream consequences of some of the laws that are being passed, some of the legislation that is being considered. So one of the long-term strategies is that. So how serious is this? Well, I would like to argue that repurposing is critical to advancing Alzheimer's disease research, regardless of whether these drugs make it to phase 3 or not. So why is that? Well, because repurposing trials are in proof of concept trials. We've heard about that. And they identify targets and pathways and combinations that can be exploited in many ways by novel compounds, by new combinations of lifestyle and drug therapy. So we're learning things about the biology of Alzheimer's disease and the biology of therapy, despite the fact that they are generic and might not be advanced to late-stage trials. The trials of generics advance many kinds of methodologies. So clinical measures. If I'll take John Harrison's neuropsychological test battery, developed by John in an academic setting, has become one of the routinely used clinical outcomes. Brain imaging. Howard mentioned the role of ADDF in amyloid PET. These things are all coming out of the infrastructure that is partially supported by repurposed agents. It's true of fluid biomarkers. It's true of digital biomarkers that we're hear about from Rhoda. It's true of trial design and analyses. We need more innovation and experimental approaches in design and analysis. And recruitment science. Pharmaceutical companies are never going to invest in recruitment science. They're going to depend on their sites to bring the patients to the site. So how do we do that better? That is an academic challenge, and one that will be supported in part by repurposed drugs. Trials of generic agents then advance the infrastructure necessary for drug development for Alzheimer's disease. This is particularly true of academic sites that are dependent on, like my site, doing the rasagiline trial, for example. Academic startups spin off from these kinds of trial sites, and they create new businesses. Very important. Lead molecules in platform trials can be repurposed agents to decrease the risk that companies feel about putting new agents into platform trials. So many examples of where repurposed agents can advance the science of Alzheimer's disease and Alzheimer's disease drug development, whether or not they ever become approved as a therapy for Alzheimer's disease. So they're a critical part of the ecosystem. Just to show you a little bit of this work that we did on rasagiline, in the lower panel here, you see the areas that had a drug placebo difference on the rasagiline patients, and you see that they had better preservation of the metabolic function in the subcortical structures and in the frontal lobes. So this was published in 2021. We're still publishing off this data set, just published a few months ago on new relationships between biomarkers. One of the things we've been exploring, for example, is the relationship between FDG metabolism on one hand, this is brain metabolism using sugar, and the formation of neurofibrillary tangles as shown by tau PET. And you can see that this is quite a compelling relationship between the two. We need to understand those sorts of things. It's done on the basis of this kind of repurposing trial. So here's the ecosystem of drug development. This is from a book that Howard Fillett and I did together. We want to keep in mind that repurposed drugs advance therapeutic knowledge. They train academic trialists and create the next generation of people who will move drugs ahead. They allow trialists to understand whether they would want a pharmaceutical career. And many of the drugs that are developed that are critical to us are developed in pharma. It supports trial infrastructure, it engages patients in communities, and it facilitates development of new therapies for many types of patients. So this is how I would summarize thinking about repurposed agents. We have Alzheimer's disease, we have a few approved therapies, and we desperately need more, right? We need more therapies for a larger range of Alzheimer's disease patients. Repurposing has a very interesting Nobel and Stockholm history in Sir James Black. Repurposing advances understanding of new therapeutics and builds research infrastructure, which is critical to advancing therapeutics in general. New strategies involve electronic medical records, computational approaches, and biomarkers, and I've shown you how that comes together to nominate new drugs for repurposing programs and other types. And the ADDF programs provide a remarkable foundation for scientific discovery and therapeutic development. Thank you very much.